Hi everyone, in this video I will be dealing with the chemiosmotic hypothesis, which is actually coming under the chapter photosynthesis, but it will be well understood only after taking this particular chapter respiration and coming in this sequence. Now this is an exact pull out from your textbook, page 214, and I am going to display what each part indicates. What is in magenta color is the thylakoid membrane. Then the space inside the thylakoid membrane is the lumen and the space outside is the stroma. So the three very important parts are marked. The pink outline, oval outline is the thylakoid membrane. And you know the specialty of the membrane, on the thylakoid membrane our ETC is located as well as the ATPase. See, our ATPase, which is indicated in yellow, is also located on the thylakoid membrane. Two things, ETC as well as the ATPase. Then the two regions where the gradient is going to be created, one is the lumen, which is within the thylakoid membrane, and outside the thylakoid membrane, that is the stroma. Clear? Now the diagram shows here very clearly 4 hydrogens or 4 protons in the lumen and only 1 proton in the outside, that is in the stroma. Now what I am going to do is I am going to make 2 very major modifications. I am going to erase all these protons, and the initial protons present in the chloroplast is going to be indicated, not the final. In the diagram it is given the final, what happens finally. So initially it is actually hydrogens or protons are more outside in the stroma. So I drew 4 hydrogens, or I wrote or indicated 4 protons in the stroma and nothing inside the lumen. Okay, so that's one major modification. Another modification is I am going to incorporate the non-cyclic photophosphorylation. Only parts of the non-cyclic photophosphorylation is shown in this picture. So I am going to incorporate the entire NCPP. And while I am incorporating it, I will just describe it once again. When the sun's light fall on photosystem 2, an electron is emitted, gets excited and emits an electron. Which is accepted by an electron acceptor. Then it passes through an ETC. See, ETC is here. Then it goes to photosystem 2. Meanwhile, the photons from the sun also excites an electron from photosystem 1, and an electron is emitted and it is accepted by another electron acceptor uphill. Then from there the electron passes downhill to this particular energy molecule NADP, and I am stopping there. Okay, so now what are the modifications I did? One, I erased all the 4 hydrogens which were present in the diagram inside the lumen, and I put those 4 hydrogens outside in the stroma. So first modification. Second modification, I incorporated the NCPP, non-cyclic photophosphorylation. Now I am going to start off. To start off, we are going to show how those 4 protons or hydrogen got accumulated in the lumen. So the first contributor is photolysis of water. As I am saying, I am going to write also the contributors of the protons, photolysis of water. So photolysis of water occurs on the inner side of the thylakoid membrane. See, the thylakoid membrane is this. On the inner side it occurs. Water is split into oxygen atom and 2 hydrogens. So instead of writing 2 H's, I am going to indicate it separately. So 2 hydrogens. So you understood how 2 hydrogens are contributed into the lumen. One is by the splitting of water. Water is split. The arrowhead should go out like this way to form oxygen atom and 2 hydrogens. Those 2 hydrogens are indicated here. So, one contributor of hydrogen or proton into the lumen is the splitting of water, which takes place where location on the inner side of the thylakoid membrane, and it is part of NCPP. Because of this photolysis only, the electron that is lost from photosystem 1 is compensated. The electron comes here. Okay, that compensated or replaced the lost electron. Then next contributor is, as I told you, the ETC. As it travels, the electrons travels through the ETC, the electron carriers remove the proton from one region and add the proton to another region. So here, which are the 2 electron carriers, B and F? There are a number of electron carriers, but they have only indicated 2 electron carriers, namely B and F. Cytochrome B and cytochrome F. Okay. So, as the electrons are passing, see, this B replaces 1 hydrogen from the stroma into the lumen. So, you saw one contributor is from cytochrome B. Then the other hydrogen is also removed. See, from by F. F is also an electron carrier. When it carries or passes the electron, it displaces the hydrogen of the stroma into the lumen. Now, did you understand how the 4 hydrogens came in the lumen? The first 2 hydrogen came by the splitting of water. Clear? The second 2 hydrogens, how did it come? One came from cytochrome B, the other came from cytochrome F. Now, I will have to tell you about these cytochrome B and F. They are part of the electron transport chain. They usually an electron transport chain carries electron carriers, but B and F, remember, are not electron carriers, they are hydrogen carriers. And therefore, because they are hydrogen carriers, when they pass the electron, they displace the hydrogen from the stroma and placed it into the lumen. So, from the stroma 2 hydrogen were displaced into the lumen. B displaced 1, so 1 hydrogen, and C displaced the second hydrogen. So, now we have 2 hydrogens out of 4 removed from the stroma. So, how many hydrogens are left now? 2 hydrogens. Now, we fulfill the lumen with 4 hydrogen. Now, we will continue with NCPP. Now, the electrons have reached the photosystem 1. Now, from photosystem 1, the electron reaches an electron acceptor, second electron acceptor, which is uphill. Then from there, the electrons goes to this substance, that is NADP. What happens there? There is an enzyme called NADP reductase, and you know the location in the stroma. Okay. On the stroma, there is the NADP reductase, which brings about the reduction of NADP with 2 hydrogens. See, from where did they get the hydrogen? The 2 hydrogens are from the stroma. So, this NADP along with the 2 hydrogens from the stroma gets reduced to NADPH plus H plus. Okay. So, as a result, the remaining 2 hydrogen, of the remaining 2 hydrogen, 1 is gone, incorporated into NADPH, and net only we have 1 hydrogen in the stroma. Finally, how many hydrogens are there inside the lumen? Inside the lumen, there are 4 hydrogens. 1, 2, 3, 4, and outside the lumen there is only 1 hydrogen. Now, a proton gradient is created across the thylakoid membrane. We have more protons in the lumen than in the stroma. Now, the gradient should break. This movement of protons from a region of high concentration to a region of low concentration through the ATPase. So, now protons move. See, the protons get in. See, I am going to transfer the protons. these protons from the lumen gets into the globular head. And what happens when these protons move into the F1 part of the ATPase, the ADP combines with P. See, ADP combines with P to form ATP. I hope you have understood how a proton gradient was created across the thylakoid membrane. Now connecting with the difference which was taken in the previous slide, you just analyze each and every step. When this photophosphorylation takes place? During photosynthesis. Where does it take place? In the chloroplast. Then where is the ETC operating? We saw it is operating in the thylakoid membrane. See, the cytochrome B and cytochrome F is shown in the thylakoid membrane. Where is the ATPase located? There, see, look at the ATPase in yellow. It is located again in the thylakoid membrane and with the F1 globular head projecting or protruding into the stroma. Then proton gradient is created, no, it is created across the thylakoid membrane. So we can say it is across the thylakoid membrane. So the two regions are the lumen and the stroma. See, lumen is inside and stroma is outside. Now again the next aspect very similar to it, where is the proton accumulated? Yes, proton is accumulated in the lumen. Next, ATP is produced where? Looking at the globular head, where is it produced? It is produced in which region? It is produced in the stroma. Lastly, energy is used by photo means light and the role of water. Now you can understand the last property, role of water. By splitting of water, what two hydrogens are contributed. So role of water is very, very important, and that splitting of water is known as photolysis. Because of photolysis, two hydrogens were contributed into the lumen, and therefore a proton gradient was facilitated. So very, very important, the role of water, photolysis of water. So once again, I am just erasing the diagram to show you the pull out from the text. So finally, I end up this chemiosmotic hypothesis with the three contributors of hydrogen. The first contributor of hydrogen is photolysis of water, splitting of water, and that is how it has contributed to the two hydrogens. See, let these two hydrogens be due to the splitting of water. So photolysis of water, which is occurring on the inner side of the thylakoid membrane. The second contributor is the cytochrome B and F, which are the parts of the ETC. These are hydrogen carriers. When electrons was passed through them, they displaced hydrogen from the stroma into the lumen. So let this be from B, cytochrome B, and let this be from cytochrome F. So now you got 4 hydrogens inside. Then next, remaining, initially we started with 4 hydrogen outside, now 2 went in. So remaining there are 2 hydrogens. How were the 2 hydrogens used? These 2 hydrogens were used in the reduction of NADP to NADH plus H plus. So finally we have 4 hydrogens inside the lumen and just 1 hydrogen outside the lumen, that is in the stroma. So a gradient is created, and to break that gradient, the protons pass through the ATPase, and the passing of the protons through the F0 to the F1 helped in the coupling of ADP to P to form ATP. So we saw how phosphorylation took place in the presence of light, and this is called photophosphorylation. I hope chemiosmotic hypothesis is clear. Thank you.